Good morning, good afternoon, and good evening, everyone. I'm Nijat Khan, Chief R&D Officer and Chief Commercial Officer at Recursion. I'm pleased to be joined by Dr. Jan Burkhardt, Principal Investigator for the Phase 2 SYCAMORE study, and Dave Morrow, Chief Medical Officer at Recursion. We're looking forward to sharing the Phase 2 data for REC-994 in CCM, which was presented for the first time yesterday at the International Stroke Conference. A couple of reminders just for context setting. First, this is the first industry-sponsored study for CCM. It is a signal-seeking study where the primary endpoint is safety and tolerability, with secondary and exploratory efficacy endpoints. And second, REC-994 was discovered as a potential treatment for CCM with the earliest version of the Recursion OS platform via phenotypic screening. And this was done via an in-licensed compound. While our platform looks significantly different today with multi-omic data layers from transcriptomic, proteomic, etc., as well as chemical synthesis that's done in-house, and large volume of patient data, just to name a few, this program represents a seed for the idea of leveraging an unbiased approach to drug discovery. Our objective today is twofold. First, on the heels of the ISC conference, to make this presentation accessible to a larger audience as soon as possible. And second, offer the opportunity to take some of your questions at the end of this webinar. As a company dedicated to advancing a diverse portfolio in areas of significant unmet needs, such as oncology, rare disease, immunology, neuroscience, and more, both through our internal portfolio and through strategic partnerships, we remain committed to a data-driven and disciplined approach, focusing our efforts and doubling down in areas where we believe we have the greatest potential to win. Win for Recursion, win for the industry, and also, of course, for the patients that we serve. We look forward to sharing more insights and progress and updates on our pipeline and partnership throughout this year. With that, I'm excited to hand it over to Dr. Jan Karl Burkhardt, to share the study data.
Hi, my name is Jan Burkhardt. I'm a neurosurgeon from Penn Medicine, and I'm excited to present today the trial results of REC-994 for symptomatic CCM patients. On behalf of all co-investigators and Recursion, I'm happy to present this phase 2 randomized placebo-controlled trial evaluating REC-994 in patients with symptomatic CCM. CCM is a small vascular malformation involving capillaries and venous structures in the brain and can present either asymptomatic or symptomatic depending on the location and the size of the lesion. Classic symptoms of these patients are stroke-like symptoms, including seizures, or can also be hemorrhage. But most of the patients are actually asymptomatic. There's no approved non-surgical therapy for these patients, reflecting that there's an unmet need for patients, especially in highly eloquent locations or patients who have more than one lesions. 360,000 patients actually have a symptomatic CCM in the US and in Europe. And of them, 90,000 patients have brainstem lesions. So there is a need for a non-surgical therapy. And if you look at REC-994, what is the mechanism of action which was investigated in this trial? You can see here on this slide an endothelial cell in a healthy patient, in a patient with a CCM lesion, as well as REC-994 impact on cavernous malformation. You can see that the CCM protein complex is actually responsible for endothelial cell activation and stabilization of the cell, which is affected by CCM. And here you can see that CCM actually decreases the cell-to-cell junction integrity and leads to the classic leakage and hemorrhage and an enlargement of these lesions. And the impact of the REC-994, you can see here that it stabilized the endothelial cell barrier function by being an active radical scavenger. So in preclinical models, REC-994 showed that the lesion size and number actually decreased by 50% and rescued also the endothelial permeability. This is here reflected on the Circulation paper from 2015 in these two graphs. If you look at our trial design, here's the screening and then randomization. schedule to 3 groups, 1 to 1 to 1. So overall, 62 patients were enrolled in this trial, into a placebo group, into a group of 200 milligrams of oral REC-994, and 21 patients in a group with 400 milligrams REC-994. These patients were observed over 12 months, as you can see here at the visit schedule in a part 1. And then now, currently in part 2, there's an extension study. So I'm just talking about the part 1 today because the part 2 is still ongoing. The enrollment criteria were MRI-confirmed CCM lesion, which was symptomatic, and the symptoms were related to that CCM. And so the patient had to have at least an MRS score of 1 or larger. And both familiar and sporadic lesions were enrolled. The study objective, the primary objective, was safety and tolerability of REC-994. There were secondary and exploratory outcomes, including MRS, patient-reported outcome, and also MRI and CT-related hemorrhagic event, and also lesion volume, hemosiderin ring changes, and biomarkers. There was no patient enrichment by symptoms or stratification in 13 US sites. These 62 patients were enrolled in 18 months. The secondary and exploratory endpoints were not statistically powered. If you look at the results, the patient demographics, you can see they were well balanced between the treatment arms. Here you can see that age, sex, race, the etiology of the CCM disease, the symptoms related to the lesion, as well as the mutations were well balanced between the groups and not significantly different. If you look at the safety, the primary outcome of this trial, you can see in the top part of this graph, any type of treatment emergent adverse events, or any symptoms a patient may have had during the time of the trial, not related to the drug itself, are reported. And at the bottom, you can see the treatment-related TEAEs. The most common adverse event, so at least 10% reported of participant, included COVID-19, dizziness, headaches, and back pain and constipation. But if you look really at like the study-related adverse events, they were only present in 7 patients, and most importantly, there were no grade 3 TEAEs presented. So that confirmed that REC-994 is safe and tolerable in these patients. One of the exploratory outcomes were MRI assessment, and here you can see the methodology for this trial. You can see that at baseline, 6 months and 12 months readings were performed, and a single blinded central neuroradiologist assessed all of these images. If you look at the data here, an exploratory analysis of the volume changes for these 3 groups. Here on the left side, you can see a waterfall plot, and you can actually see every single patient enrolled in this patient with the volumetric changes. You can see if the graph is going up on the graph in the positive, that means the lesion increased over the 12-month observed period. If the graph is heading down, it means the lesion size decreased. On the right side, you can see the absolute numbers, and you see that really like REC-994 in the 400 millimeter group decreased the absolute mean change in lesion volume per patient to 457 cubic millimeters compared to an increase in the placebo and 200 milligram group. Also at the bottom, you can see that the percentage of patients with any type of reduction in lesion volume decreased by 50%, and 50% of the patients in the 400 millimeter group compared to 28 and 24% in the 200 milligram and placebo group. If you look now at clinical secondary exploratory outcome based on MRS score, as a reminder, MRS1 was an inclusion criteria, so that's why at baseline, every patient has at least MRS1 or higher. And on the right side, you see the 12-month follow-up data, and here you can see this. If you look at the 400 milligram group, there's a trend towards improvement and stabilization of the symptoms, while you see in the placebo group, there's a trend towards worsening of the symptoms, which is expected as a natural history. Again, these analyses were not powered, but show a nice trend towards efficacy of REC-994 in the 400 milligram group. If we now look at another subgroup analysis of this, so only patients who had a brainstem cavernous malformation, and these are exactly the patients, as I mentioned in my introduction slide, who would benefit from a medical therapy, giving the complexity for surgical resection with a high chance of comorbidity during surgery. So these patients would really benefit from a non-surgical therapy. And here again, you can see the effect in the 400 milligram group with an absolute mean change in lesion volume per patient of 256 compared to the control and the 200 milligram group. And again, here, 50% of the patients showed any reduction in lesion volume after 12 months in the 400 milligram group. Similar to the MRS, you can see here too, you can see the changes similar to the overall group. The brainstem lesion had actually here in the 400 milligram group, you can see a trend towards improvement of the clinical symptoms after 12 months versus the placebo group who showed worsening over time. This is a case example which nicely puts everything together where we talked about the clinical and MRI findings. It's a 39-year-old with a devastating large brainstem cavernous malformation. Here you can see the baseline image. She presented with severe, moderate to severe CCM symptoms, including mobility issues, hearing loss, headaches, and blurry vision symptoms. And after 12 months with daily 400 milligram REC-994, you can see a volume reduction of 38% of the lesion and also complete resolution of her symptoms. And this is just a screenshot showing a 2D image. This was obviously based on a volumetric 3D analysis. We did also additional analysis, which is a little bit too much for this presentation in terms of timing, but we want to mention that there was also a trend seen in hemosiderin ring reduction in the 400 milligram arm, as well as seizure frequency appeared to be reduced in the 400 milligram arm, but there was an imbalance with respect to seizure history across the arms, so it's hard to tell if this is really a true effect or if, and we need to study that more in the future. The incidence of new symptomatic hemorrhage events were comparable across all the arms, and the patient-rated outcomes did not demonstrate any differences between the treatment arms of the study or placebo. So in conclusion, these phase 2 results were able to show that REC-994 was observed to be safe and well tolerated with no treatment-related grade 3 or larger adverse events. The REC-994 400 milligram treatment group reduced lesion size and volume in CCM patients, including those with brainstem lesions, and also reflected that the clinical symptoms showed a trend based on the modified ranking score that 400 milligram patient group improved clinical function at 12 months, including the patients with brainstem lesions. So the next steps are there's a long-term extension, part 2, and it's ongoing, and we're really excited to see what these results will show us. I want to thank again all the investigators and the sites enrolling in this exciting trial, and most importantly, we want to really thank all the patients and their families to participate in this trial and the Alliance to Cure Cavernous Malformations for their support. Thank you so much.
Thank you, Dr. Burkhardt, and to the entire team. I just want to echo something that you mentioned, which is thanking the patients first and foremost, their families, the Alliance for Cures, and of course, our investigators for this great effort. A couple of next steps. First, Recursion intends to submit this data for publication in a peer-reviewed scientific journal. And in terms of next steps for the program, this will be guided by the upcoming regulatory discussions as well as the ongoing long-term extension study. With that, I'd like to introduce Dr. David Morrow and have him join me to answer some of the questions that we've received so far.
Good morning, everyone.
Thanks for joining, Dave. I'm looking through the questions. Let's see. All right. First one, we're getting quite a few questions on the modified Rankin score. So, and I'll take the one from Eric Joseph from JPM as well. Why is the MRS Rankin score important? Why is it important to measure versus other? Is the MRS a sensitive enough measure at the lower end of the scale, so scale 0.2 or below, to significantly detect differences in functional outcomes? And if MRS3 is considered a meaningful threshold in patient function, what would be viewed as a clinically meaningful treatment effect? So I'll kick it off, and then Dave, I'll hand it to you for additional comments. Just in terms of the modified Rankin score, in discussions with neurologists, this is a clinician-reported endpoint and outcome that's been highly recommended. First of all, it is a global measure, and it's known to be a global measure for functional disability. The scale goes from 0 to 6. It has been used previously in acute stroke trials as well, in terms of a measure as to how the patients are doing. There are both in pivotal studies, I would say, and then also in approved products. The other thing that's important to note here is, in terms of the improvement and stabilization. So for chronic diseases, one of the topics that's come up both from patient advocacy groups and from neurologists is across the scale, any change is considered clinically meaningful, and as well as stabilization for those patients. So those are a couple of the points, much more to do. But Dave, additional comments from you?
Yes. Thank you, Najat. In a lot of conversations we've had to really understand the relevance of the Rankin score for patients with CCM, one of the consistent comments that we've gotten from neurologists as well as patient advocates is that these patients' MRS tend to become worse over time. So as Najat mentioned, stabilization is going to be key as well as improvement. So, as we look at the data, we look at sort of two elements of the data. How much are patients improving and how much are patients stabilizing over time? Given that these patients live with the disease for decades, we think improvement as well as stabilization is going to be important. As far as sort of moving sort of down the scale, what we've understood is that any improvement from a 3 to a 2, et cetera, is important. And we think that there is enough precedent and use with MRS amongst investigators as well as experience with regulatory authorities that it is a sensitive enough scale to evaluate efficacy of a therapy.
Thank you, Dave. And yeah, I mean, methods such as the dichotomous approaches and others are also in place that has precedence here as well. All right, I'll go to the next question. Again, quite a few questions on brainstem subpopulation. So in the deck, you had a breakout of subsets of patients. Tell us more about the significance of brainstem subpopulation. So just a couple of points to note. We said that we looked at the symptomatic patient population for CCM. About 1 out of 4, and that equates to around 90,000 patients in the US and EU have brainstem lesions. In discussions again with neurologists, a couple of things to note. One is brainstem lesions, even a couple of millimeter change, has pretty significant consequences in terms of functional ability and outcomes for patients. So lesion volume actually becomes, and size becomes important there. Second is also challenges in terms of surgical procedures for patients with brainstem lesions. And there's also been studies done in parallel looking at some of the top predictors for rehemorrhage and rebleed events, and patients with brainstem lesions, lesion volume are considered among the top 2-3. So just a few things to note there, but I'll leave it to Dave to add more color.
Yeah, we also looked at many other subpopulations, and we'll be showing in subsequent manuscript also tended to favor the 400 mg arm. So it wasn't just restricted to brainstem, but obviously, the patients with brainstem lesions are without a doubt the most problematic. And in fact, most centers in the US that participated in the study really have no options, really just sort of observation. These are patients that have the most morbidity, they have the most severity of disease, they have a greater incidence of hemorrhage. So clearly, if there is a therapy that can reduce lesions and also improve symptoms, not only for brainstem patients, but for all patients, we think that this could be a real meaningful improvement in the treatment of patients with CCM.
Thank you, Dave. And I'll go to our next question. This is from Alec from Bank of America. What does the company think about the registration path from here? Would impact on disease from the long-term extension be enough, or would, say, a phase 3 likely be required? And also sub questions to it, which is around, given the tolerability at the 400 milligram dose, would further dose escalation be of interest to drive greater lesion reduction? Or are you satisfied with what you're seeing at 400 milligram? These are great questions, Alec. I think for us, a key next step is engaging with regulators, of course, as we mentioned, and we're doing that in the upcoming 1 or 2 quarters, as well as looking at the long-term extension results. From what we understand, based on that, and again, the conversations that we will have, is a pivotal study will likely be needed. But I'll hand it over to Dave to add more color.
Yeah, the only other comment that I'll add, Najat, is this is the first time the FDA will ever see any phase 2 data from a CCM. We anticipate that they will be collaborative and they will work with us to really define what is the best path forward. Unfortunately, there is no regulatory precedent for CCM, but if you look at sort of elements, we think that reduction in lesion volume is important, and we've talked about MRS being a prior endpoint that has been accepted by the FDA. So that, along with additional data from LTE, will inform our discussions with the FDA, and hopefully we will collaborate to develop a path forward.
Thank you so much, Dave. Yes. So in a nutshell, more work to be done, and stay tuned. But as always, across the portfolio, we'll be data-driven and disciplined in our path forward, doubling down on areas where we can win. So with the patients at the center of everything that we do. So with that, just want to thank everybody again for joining our webinar today. Thank you for taking the time. The presentation and a recording of this webinar will be made available on Recursion's IR site shortly after we sign off. Thank you, everyone, and have a good rest of your Thursday. Bye.
Thank you.